So folks, the other part of this lesson, because the title was Dihybrid Crosses and Other Types of Dominance, is to introduce you to the idea that complete dominance, which is one of the things Mendel studied, is not always the scenario that happens. So I have a picture with flowers that we'll do some crosses with. I also have a hilarious picture that shows a regular incandescent light bulb and an ice cream cone that have gotten married and produced a child who's an LED light bulb, and it's supposed to be funny. But the reason I put it here is because that picture kind of illustrates what incomplete dominance is. Incomplete dominance is when neither allele can mask the other. In complete dominance, one allele totally masks the other. What that means is the dominant allele just showed up. The fact that the person had a recessive allele really didn't make a difference for their phenotype. But in incomplete dominance, now it's going to make a difference. Neither allele can mask the other. The result is that you get an intermediate trait. So up until now, when we were looking at a gene, there were only two possible phenotypes, the dominant one or the recessive one. Now we're going to add a third phenotype in, the intermediate between the two alleles that we have. So you can see in my example that we're going to look at pink and red and white flowers. So I need to remind you that in kindergarten you learned about colors, and that you should be comfortable understanding that that blue and yellow make green, that blue and red make purple, that red and yellow make orange, that big and small average out to medium, that general idea, okay? Now we're going to use this 4 o'clock flower plant, that's the plant that's in the picture, to illustrate what incomplete dominance would look like. So you can see over here I have my generations labeled as P and F1 and F2. In my parental generation, I have a red flower and a white flower, and all of their offspring are pink. That's weird because neither parent was pink. So how on earth were all of their offspring a different color than the parents? Well, my explanation would be that all of the offspring must have gotten one red and one white allele. Red plus white makes pink color-wise. So if you ever see a cross where you have two parents who have two opposite traits, and then the offspring all have a third different trait that neither one of the parents had, that's a really good indication that it might be incomplete dominance, especially if the offspring have a trait that's in between the two parents' traits. In this case, pink is like a mixture, an intermediate between red and white. Now the way that we represent crosses won't change, right? We're still going to make a Punnett Square, but the way that we write the alleles will. Now remember, this is just strategy. The actual letters that you're picking are not super relevant. But here is one way that you could represent this. Red and white, neither one of them is dominant over the other. Neither one masks the other. So it doesn't really make sense to use a capital and a lowercase letter because one of them isn't dominant and the other one isn't recessive. So in this scenario, typically what we do is we use just capital letters, or if you like lowercase letters, you can use just lowercase letters, whatever floats your boat. But that we would do something to indicate how they were different. So for example, if I'm going to cross a red with a white flower, I might represent the red flower's genotype like this. The red flower, I'll do it over here where I have a bit more space. I'm going to use the letter C for color because I don't want to use red or white. Neither one of them is dominant. And I'll put 2 1s to represent red. Now sometimes you'll see this with numbers. Other times you might even see it with letters. So maybe there'll be an R here instead of a 1. what I hope you're seeing is that it's not super important whether I'm using a number or a letter, but I'm doing something to differentiate these color alleles from these. So I could put a 2 for white, or I could use the letter W. I like to use letters because then I remember what they are. Sometimes you'll see examples with numbers, and I find that when I use a number, I always have to go back and be like, what was number 1 again? I don't even remember. But when I use a letter, I'm like, oh yeah, W, that was white. So notice, I'm just representing things differently. Now, of course, the way you solve a problem is up to you. And sometimes the actual letters you pick is not relevant, or are not relevant, but this is pretty common for how you would see me represent incomplete dominance. I'll use capital letters, and then I'll use a superscript to distinguish between the two. So in my parental generation, when I cross that to make the F1 generation, this is what my Punnett square would look like. I'm crossing a red flower with a white one. Now, how did I know the parents were homozygous? Well, since I can see that there's an intermediate color, this pink color, my conclusion is to be pink means to have one red and one white allele. That's what I'm inferring. So for that flower to be red, it must not have a white allele, and vice versa for the white flower. So that was how I would have inferred this in this question. I'm telling you so that you know. Since both parents are homozygous, I kind of don't need those parts of my Punnett square because I'm just repeating myself. So there's really only one option for the offspring. All of the offspring in the F1 generation will have one red and one white allele. The phenotype that this gives is pink. And if you look at my picture, that's exactly what it shows. All of the offspring in the F1 generation were pink. And the way they were pink was because they have one of each of these alleles. Now in the next Punnett square, we're going to take the F1 generation and make an F2 generation. When we're doing that, unless it specifies otherwise, you always take two F1 organisms, they generally have the same genotype, and cross them. So I'm going to take two of these pink flowers and cross them. The pink genotype was one white and one red. And notice I wrote them in a different order because it didn't really matter. Red wasn't dominant over white, white wasn't dominant over red. So I didn't have to put the capital letter first like I usually do because they're all capital letters. So it didn't matter which one I wrote first. What does matter though is how I fill out my Punnett square. Since my parents in this particular cross are both heterozygous, they can each make two different gamete possibilities. They could make a gamete with the white allele, or they could make a gamete with the red allele. So when I fill in my Punnett square, I will get a box where I have homozygous for the white allele. I'll get two boxes, this one, where I'm heterozygous, I have one white, one red. And I'll get a box where I'm homozygous for the red allele. Now remember, your Punnett square isn't your answer, it's just your work. What this tells me is that this one would be white, this one would be pink, this one would be pink, and this one would be red. When incomplete dominance is the scenario, being heterozygous means that you have an intermediate trait. So it's different than complete dominance. Now if you look at these proportions, I have one white, two pink, one red. And that's exactly what's shown in that picture right above. So the ratio of white to pink to red would be 1 to 2 to 1. Now when we talked about complete dominance, I said that when you cross two heterozygous organisms, 3 to 1 is the ratio you expect. 3 with the dominant phenotype, 1 with the recessive. This is the ratio that you expect when you cross 2 heterozygous organisms where we have incomplete dominance. 1 to 2 to 1. 1 of each homozygous trait, and then 2 of the intermediate trait. Now the way you'll recognize incomplete dominance is either the question will flat out say it, these alleles are incompletely dominant, or there will be 3 phenotypes, 3 choices, because in complete dominance there are only 2 choices. Now if you would like to ask a question about this, I feel like I want some ice cream right now. I keep looking at my ice cream cone picture. Yeah.
What kind of question would this be on a test? Like, would it be numerical response?
Most of the questions will be multiple choice and numerical response. So most of the time, the question will be asking you something about how many of the offspring you expect to have each trait. Sometimes the questions ask you to work backwards, like we've done before, where I would say, here's what the offspring are like, what did the parents have? All of the practice we're doing is written questions so that you can get in the habit of doing all of the steps. But most of the test questions will be multiple choice and numeric response, where you're just picking your answer. For numeric response, it's usually ones like this where I'm asking you like what percentage or what ratio you expect in the offspring. Okay, so here's a people version. Oh, sorry, I can go back.
Sorry, I'm just
My bad. It's okay, I can keep looking at that ice cream cone and thinking about where I can go get an ice cream cone and when I'm gonna do that. I'm interested to know, do you guys have an opinion? Dairy Queen or McDonald's soft serve? Which one do you prefer? Let's have a vote. How many people think Dairy Queen? Oh yeah. How many people think the McDonald's one? I pick that one because it's so much faster and cheaper. And I feel like those outweigh my other characteristics that I vote for. And whoever's watching this video has now heard my thoughts on soft serve ice cream. Okay, so here is a real life person example of an actual trait that humans can have that shows incomplete dominance. It's for a disorder called sickle cell anemia. Maybe you remember hearing about this in Bio30E. It's when your red blood cells are the wrong shape. They're kind of crushed looking instead of nice round life-savory looking things. There are 2 potential alleles that a person could have that affect sickle cell anemia. This HB stands for hemoglobin, and like most human traits, we're using 2 letters to represent 1 gene because there are not enough letters in the alphabet for all of the genes that we have to represent. So this HB is one thing. It's not 2 different alleles. And then you'll see that one of them has an A as a superscript and the other has an S. The S refers to the sickle trait. Now sometimes the intermediate trait that you get when 2 alleles are incompletely dominant is indifferent, like with color. You're red, you're pink, whatever. Is that really helping you survive? Maybe, but maybe not. But in the specific case of sickle cell anemia, heterozygotes have an advantage. Not only do they not really have sickle cell anemia, but they're also more resistant to malaria than people who are homozygous either way with this trait. So, in this question, we have 3 possibilities. We have a person who is normal, who is malaria resistant, or who has sickle cell anemia. Those are our 3 phenotypes in this scenario. So a person who's normal, who doesn't have the sickle cell anemia and doesn't have malaria resistance, would have 2 copies of the HbA allele. A person who has sickle cell anemia will have 2 copies of the S allele. And a heterozygote means you have 1 of each.
What does the HB stand for?
Hemoglobin.
How do you spell that?
Hemoglobin. Hemoglobin. It's part of your red blood cells is where oxygen attaches. So the cross I'd like us to do is this. So you take a moment and you do this cross. Decide what the genotypes of a normal and a resistant person are. So that's malaria resistant. And let's figure out the phenotypic ratios in their offspring. So folks, the first thing I would do is fill out what the parent's genotype is. That's always the first thing that I do. Even if I don't know all of the answers, I always start there. The person who is normal, isn't malaria resistant, doesn't have sickle cell anemia. So they must be homozygous for the normal allele. So I wrote HbA as their genotype. Remember that this is all one thing. We just need 3 letters to represent it because there are so many different human genes that we need to represent. The person who's malaria resistant would have 1 copy of the normal allele and 1 copy of the sickle cell allele. So when I go to make my Punnett Square, I'll say, one of my parents is homozygous. That means there's really only one option for what their gametes can look like. So for the parent who had 2 copies of the A allele, I could write it twice, but that seems silly, so I'm not going to. For the parent who had 1 copy of each allele, they can make 2 possible gametes. And so I'd write both possibilities. That means that in their offspring, I would expect 50% to have 2 copies of the normal allele and 50% to be heterozygous. So when I report the ratios in my offspring, I could say the ratio is 1:1, normal to resistant, if I'm giving a ratio. Or I could say 50% normal, 50% resistant. Now there's a third phenotype in this question, the sickle cell anemia. No one in this predicted outcome would have sickle cell anemia, but you might have to report that as part of your answer. So it's possible that there's a third spot for you to fill in particular in a numeric response question. And you might have to put a 0 because you might predict that none of the offspring should have that phenotype. That won't always be the case, but just know that's a possibility, that 0 of the predicted offspring will have a phenotype, and that you'll have to fill in a 0 in the numeric response. Anyone want to ask anything about this example? Okay, we got one more then. There's another scenario, and it's called codominance. And sometimes people think incomplete and codominance are the same because they both involve stuff kind of mixing together. But there's a distinction. In codominance, both alleles are fully expressed. That means that they're not mixing together to form a third intermediate color. It means that we're going to see both traits. So we're going to use roan horses as our example here. From far away, you might not be able to tell, but a roan horse actually has 2 different colors of hair, usually one light color and one dark color. The exact shades can vary from black to brown to red to beige to white, whatever. But the idea is that a roan horse has 2 different alleles and they both show up. So in our last scenario, incomplete dominance, if I mixed black and white, I should get gray, right? That's the intermediate. In this scenario, if I mix black and white, I get black and white. Both colors show up. So I might represent this roan horse's genotype as a C for color. That's my standard color representation. But the actual letter you're choosing is not the most important thing. I put a superscript to indicate that there are 2 different alleles. And they're all capitals because neither one of them is dominant over the other. B stands for black, the W stands for white. And what we're saying is, this horse isn't gray. What this horse actually is a horse that has black hairs and white hairs. Both alleles are fully expressed. Now, the cross that we're going to do is two roan horses. So here's the genotype for a roan horse. I'd like to know what the ratio in the offspring you would expect is. And then as a secondary question, could we have pure breeding roan horses? That's a yes or no type thing. So I'll pause here while you think about the Punnett Square. So folks, hopefully your Punnett Square looks or is looking like mine. The fact that I wrote the black allele first and the white allele second is not relevant. I could easily have reversed the order and I would end up with the same answer. If I fill in my Punnett Square, I will end up with one of the four squares giving me two copies of the black allele in the offspring.
Is the roan just black and white?
Correct.
So it'd be a mix like zebra.
But not as distinctly striped. The black and white hairs are like all over the place.
Or something.
Yeah.
Kinda
This is a roan horse right here. Where like from far away, you can't even sometimes tell that there are two colors. But if you look up close, there's different hair colors right next to each other. So out of those four squares, one of them would give me two black alleles. So I'd have a black horse. Two of them, let's color code things, this one and this one, give me one black and one white allele. And we said right from the beginning, that's what a roan horse was. And then another box down here gives me two white alleles. That gives me a white horse. Doesn't that ratio look a lot like the flower ratio, 1 to 2 to 1? It's the same ratio. Our Punnett Square looks basically the same. We used a letter with a superscript and we changed the superscript. The only thing that's really different between codominance and incomplete dominance is what exactly this third phenotype is. In incomplete dominance, the third phenotype is a mixture between the two others. It's intermediate. In codominance, the third phenotype is what you get when you have both of the other phenotypes at the same time. A roan horse is essentially black and white. Recognizing the three different dominance scenarios, complete dominance, incomplete dominance, codominance, is a really important thing. And that's something that there could be a question about where you're not even predicting offspring, but you're just explaining how those three types of dominance work. Now, that is the end of the notes here. The homework for today is the other part of lesson 2 on your practice problems sheet. So it lists a few more questions, a little bit from the digital textbook as well, specifically one asking about the types of dominance.